Welcome to the deep dive. Today, we're really getting into the weeds of virology, taking a high-yield look at the viral playbook.
Right. We've got this stack of sources on structure, replication, latency, all that good stuff.
And the mission is to pull out those core ideas, the rules that govern how these things work, right?
Yeah, exactly. We want to understand the why. Why is one virus super fragile, needs close contact, while another can survive, like a trip through your stomach?
Yeah, why soap works on some but not others.
It often comes down to structure, stability, transmission, persistence. It's all linked. We're decoding the viral rules.
Okay, so let's start right there. That structural difference seems key. Naked versus enveloped, why does it matter so much?
It's absolutely fundamental. It dictates how tough a virus is outside the host. Let's talk about the enveloped ones first, the fragile ones.
And these are the ones that kind of wrap themselves in our own cell material.
That's right. They acquire this lipid bilayer, basically a piece of host and cell membrane, when they bud out.
From the main cell surface, or?
Could be the plasma membrane, or sometimes the nuclear membrane, even Golgi, like with herpes viruses. They essentially steal a coat on their way out.
So they've got this host-derived lipid coat.
Precisely. And that coat, well, it helps them get into the next cell, but it's also their Achilles heel. Lipids just don't stand up well to the environment.
Ah, so detergents, like soap.
Soap, heat, drying, acid, even bile salts in the gut. They disrupt that lipid envelope pretty easily. Think of it like an armored car versus one with maybe just a tarp thrown over it.
Okay, so the tarp gets ripped easily.
Yeah, the envelope pops, which is why enveloped viruses like HIV, influenza, HSV, they generally need close contact.
Right, fluids, droplets, that kind of thing. They can't just hang out on a surface for days.
Not typically, no. They're just too fragile compared to the alternative, the naked viruses.
The non-enveloped ones, they skip the coat.
They did. They're just the protein nuclear capsid. It's a much tougher structure. Think protein shell versus lipid bubble.
So they can handle the rough stuff.
Absolutely. Acid, detergents, drying, bile. They're much more resistant.
And that's why they can survive the GI tract.
Exactly. That structural stability is what allows for fecal-oral transmission.
Like norovirus, Hep A, rotavirus, like classic stomach bugs or foodborne viruses.
Yep. They're built tough for environmental survival and passage through the gut. That simple structural difference explains a huge amount about how different viruses spread.
Okay, crystal clear on structure and survival. Let's shift gears to replication. Some viruses seem to play by different rules, especially the DNA viruses. We usually think nucleus, right?
Usually, yes. DNA viruses typically need the host cell's nuclear machinery, the polymerases and stuff, to replicate their genome. But then you have the pox viruses.
The big exception, like molluscum.
Exactly. They're these huge, complex, double-stranded DNA viruses, but they do everything out in the cytoplasm. They completely ignore the nucleus.
How do they get away with that? Don't they need the host's DNA tools?
Well, because they're so big, they basically bring their own factory. They encode and carry their own DNA polymerase, RNA polymerase, all the essential machinery.
Wow, so they're self-sufficient almost, setting up shop right in the cytoplasm.
Pretty much. It gives them an edge, lets them bypass some nuclear defenses.
Okay, that's a cool DNA anomaly. What about weirdness on the RNA side, or maybe blurring the lines? I'm thinking Hep B.
Hepatitis B. Yeah, that one's fascinating. It's technically a DNA virus, a hepatovirus, partially double-stranded.
But it uses that enzyme we normally linked to retroviruses.
Reverse transcriptase, exactly. During its replication, it actually makes an RNA intermediate and then uses reverse transcriptase to make new DNA copies from that RNA template.
So it borrows a trick from HIV's playbook, essentially.
It uses the same type of enzyme, yes. But a key difference is that HBV doesn't typically integrate its DNA into the host chromosome like HIV does. It's just part of its replication cycle. Still, very unusual for a DNA virus.
Nature really likes to reuse tools, huh? Okay, from weird replication to, well, chaos. Mutability. How does a virus's genome structure make it more likely to cause, say, a pandemic?
Yeah, genome organization is critical here. The real game changers are viruses with segmented genomes. Their genetic material isn't one long strand, it's in multiple pieces.
Like chapters in a book instead of one scroll.
Kinda, yeah. The mnemonic we use is BIA, Bunya viruses, orthomyxoviruses, arenaviruses, and rheoviruses.
And orthomyxvirus means influenza.
That's the big one.
Yeah.
Because the genome is in segments. If two different strains of influenza A infect the same cell, say a human flu and a bird flu.
Their segments can get mixed up.
Exactly. They can swap segments during packaging. It's called antigenic shift. You get this dramatic reassortment, creating a completely novel virus subtype.
Which our immune systems have never seen before.
Precisely. Zero pre-existing immunity in the population. That's what triggers pandemics. Big, abrupt changes.
Okay, that's shift the big jump. What about the smaller yearly changes in flu? Or viruses like Hep C that are just constantly changing?
Right, that's antigenic drift. It's due to small, frequent point mutations. RNA viruses especially tend to have error-prone polymerases. They make mistakes when copying their RNA.
Like typos in the genetic code.
Lots of typos. Think about hepatitis C virus, HCV. It's a positive-sense RNA virus, a flavivirus. Its RNA polymerase is notoriously sloppy.
So every new virus particle might be slightly different from the parent?
Often, yes. Especially in the envelope proteins, the parts the immune system sees. So the antibodies you make against version 1.0 might not work so well against version 1.1, which pops up minutes later.
And that makes it hard for the immune system to ever get ahead.
Extremely hard. That constant mutation is why HCV evades clearance so often. Something like 60 to 80% of infections become chronic. The immune system is always playing catch-up. It's a brilliant survival strategy, evolutionarily speaking.
Wow. Okay, that constant change is one survival strategy. Let's talk about another, just hiding out. Persistence. The herpes viruses are masters of this, aren't they?
Oh, absolutely. The herpes viridae family, they're enveloped, double-stranded DNA viruses. They wrote the playbook on latency, lifelong tenants.
So they infect you, cause an initial disease maybe, and then vanish.
They retreat. They go dormant in specific, safe locations in the body, often nerve cells where the immune system has a harder time reaching them. They just wait.
And then pop out later under stress or if immunity wanes.
Exactly, reactivation. And they have very specific hiding spots.
Let's run through those key ones. HSV-1, the cold sore virus.
That one establishes latency primarily in the trigeminal ganglia, nerve cluster near the temple.
Okay. And HSV-2, genital herpes.
That typically sets up shop in the sacral ganglia, down near the base of the spine.
Makes sense location-wise. What about chickenpox shingles? VZV.
Varicella zoster virus, HHV3. That hides out in the dorsal root ganglia all along the spinal cord.
Hence, shingles often following a nerve distribution.
Precisely. Then you have the big ones that infect immune cells. EBV, Epstein-Barr virus, HHV4.
Mono, that one likes B cells, right? Which is linked to some cancers.
Yep, latency is in B cells, and yes, it's associated with things like Burkitt lymphoma. And then there's CMV, cytomegalovirus.
HHV5, another cause of mono-like illness.
It can be. CMV establishes latency in monocytes and macrophages. Immune cells again. That's the one famous for causing those owl's eye inclusions inside infected cells under the microscope.
So they all have their preferred hideouts. Amazing persistence. Let's talk about another virus famous for persistence, but in a different way, HIV. Why is lifelong therapy, RT, needed even if the virus is undetectable?
Ah, the latent reservoir. This is the huge barrier to an HIV cure. Our drugs are fantastic at stopping the virus from replicating now.
They block enzymes like reverse transcriptase integrase.
Right. They stop new virus production and prevent infection of new cells. So the active viral load in the blood plummets, often to undetectable levels.
But the virus isn't truly gone.
It's not gone. Because HIV is a retrovirus, it integrates its genetic material as DNA, called a provirus, directly into the host cell's own DNA.
Permanently stitched into our chromosomes.
In some cells, yes. And crucially, it does this in very long-lived memory, CD4, plus T cells. These cells can enter a resting state.
So the cell isn't actively making virus.
Exactly. The integrated HIV provirus is silent, transcriptionally inactive. Because the cell isn't making viral proteins, it's invisible to the immune system. And because the virus isn't replicating, RT drugs have no effect.
It's completely hidden, dormant, inside our own cells.
Correct. And these cells form a stable, long-lasting reservoir. If someone stops RT,
those cells can wake up, reactivate.
the provirus gets transcribed, new viruses are made, and the infection roars back very quickly. Viral rebound.
And is there a primary location for this reservoir?
The largest concentration of these latently infected cells is believed to be in the gut-associated lymphoid tissue, the JALD. It's a huge immune organ lining the intestines.
Okay, so that's the long game challenge. But quickly, what happens if ART isn't working well or isn't available and the CD4 count drops? Those classic opportunistic infections start appearing at certain thresholds, right?
Yes, the CD4 count is a critical marker of immune health. Once it drops below 200 cells per microliter.
The big worry is PJP.
Pneumocystis pneumonia. Exactly. Classic signs are often a non-productive cough, shortness of breath, hypoxia, and you see those bilateral ground glass shadows on the chest x-ray. Standard treatment and prophylaxis is TMP-SMX.
Try methoprim sulfamethoxazole. Okay, what about below 100?
Below 100, you get really concerned about CNS infections, particularly Toxoplasma gondii. That typically shows up on MRI as multiple ring-enhancing lesions, often in the basal ganglia.
Got it. And there's also a viral cancer we see.
Yes. Kaposi sarcoma caused by HHV8 usually appears when the CD4 count is below 200, sometimes a bit higher. Causes those distinctive purple or reddish-brown vascular skin lesions, but can affect internal organs too.
Wow. Okay, we've covered a huge amount, from basic structure enveloped versus naked, determining stability and transmission.
To the weird replication of pox in the cytoplasm and HPV using reverse transcriptase.
Then the segmented genomes causing antigenic shift in flu and the constant drift in HEV.
Followed by the specific latency hideouts of the herpes viruses.
And finally, the massive challenge of the latent HIV reservoir in resting T cells, mainly in the GALT.
And the common thread is that understanding these fundamental viral strategies, structure, replication, latency, mutation, is key. It explains why some viruses spread easily, why some persist for life, why some treatments work, and why others, like an HCV vaccine, are so difficult to develop, while an HBV vaccine is highly effective.
It all links back to the basic biology.
It really does. Which leads to maybe a final thought.
Go for it.
If the major hurdle to curing HIV is this silent, latent reservoir, especially in those GALT memory T cells, what's the winning strategy going forward? Is it going to be finding clever ways to shock and kill, wake those cells up so our immune system can eliminate them? Or is it finding a way to enforce permanent latency, a block and lock strategy, to ensure that integrated provirus never reactivates, even without daily art? Which path holds more promise? That's a huge question in biomedical research right now.